Hi everyone, we'd like to extend a warm welcome to all the fertility clinics, genetic counsellors and other clinicians that have joined us for the webinar this afternoon in what can only be described as a very challenging time. I'm Kelly Lochenberg, the genetic counsellor at Next Genetics. I think I've met most of the people who are joining us this afternoon at various education events that Next Genetics has hosted. I'm sure you're all very familiar with Next Genetics and the products and services we offer along the reproductive journey. As you know, one of our main services is preimplantation genetic testing using next generation sequencing technology. This has been offered since around 2016. So we're very excited to be bringing you this webinar today in conjunction with Vitrolife, who supply us with our PGT-A kits. We're very privileged to have Prof. Alan Handyside presenting today, who, as you're aware, is a pioneer in the field of preimplantation genetic testing. Prof. Handyside has previously been director and consultant in preimplantation genetics at the Bridge Clinic in London for 20 years, principal scientist at Illumina, and is currently a scientific advisor in genomics for Vitrolife. Prof. Handyside will be presenting an overview of the current literature and trends in the field of PGT-A and give his own perspectives of the future of this field. Before I hand over to Prof. Handyside, just to let you know that you're welcome to type questions into the question box during the presentation. At the end, there'll be a Q&A session where Prof. Handyside will then answer these questions depending on time. So without any further delay, thank you so much, Prof. Handyside. We look forward to hearing your presentation.
Well, thank you and welcome everyone, particularly my friends and colleagues in South Africa. It turns out I have family in South Africa and a couple of my cousins are out there with you at the moment. So this is going to be a Vitrolife webinar updating you on pre-implantation genetic testing for aneuploidy. Today I'm going to focus particularly on using PGT-A, as it's now known, as an additional embryo selection tool, and share with you my own experience of setting up an IVF program dedicated, in our case, to single embryo transfer for all patients, good and bad prognosis of all ages. So I'm going to begin by reviewing exactly where we are today, by showing actually some the latest data from the Japan registry. And of course, Japan has very much focused on single embryo transfers and pioneered the cryopreservation technique of vitrification. So I'll be showing some of their data from 2017. I'll then briefly review the genetic basis for doing PGT-A and review two recently published reports of experience from two clinics. The first of those clinics is based in Southern California and actually is the summary of over 10 years experience using PGT-A for all patients. And the second will be our own, my own experience as an option for patients having single embryo transfer. I'll then share what I consider are the main benefits of including PGT-A for embryo selection and provide some thoughts on how to set up your PGT-A program in your own clinic. Finally, I'll highlight some exciting future trends. So I think there's always room for improvement. So to begin with then, I start with these plots of IVF success rates plotted against maternal age. So historically, 40 years ago, at the beginning of IVF, all normally fertilized embryos were actually transferred together. And yet at that time, pregnancy rates were very low, between 5 and 10%, even in younger, good prognosis women. However, with progressive improvements in ovarian stimulation, culture media, and lab standards, pregnancy rates in the late 80s and 90s improved dramatically, requiring the number of embryos transferred to be reduced to avoid higher order multiple pregnancies. And now throughout the world, it's one of the main goals of IVF to move, as some Scandinavian countries have done through regulation, to single embryo transfers to minimize multiple pregnancies, with, of course, their associated risks for both patients and children. So what we see here from the registry for 2017 is primarily fresh and frozen single embryo transfers. A majority are frozen. And in Japan, there's only a small amount of PGT-A at the present time. So this, if you like, gives us a baseline, and I'm going to use this information all the way through when we look at the effect of adding in PGT-A. So if we just look at pregnancy rates per transfer, and these are primarily, as I've said, single embryo transfers, up to the age of 35 now, even with a single embryo transfer, we get remarkably good pregnancy rates. But beyond the age of 35, these rates come down really quite dramatically towards the, women in their mid-40s. And so treating patients in this age range is much more of a challenge. You can see that obviously there is some loss through pregnancy. Anomalously, these very young patients actually appear to have a higher miscarriage rate per pregnancy. But the loss rate in younger women tends to be around between 15 and 20%, so that the live birth rate per primarily single embryo transfer comes down along this line. However, because single embryos are being transferred, the multiple rate remains very low, between 3 and 5%. However, once you go beyond 35, the miscarriage rate comes up very sharply just at the same time, and we now know that much of this is due to chromosomal aneuploidy, and indeed the main cause of this are errors in female meiosis, so that the eggs are actually aneuploid. And for that reason, those embryos, when they're fertilized, are actually not viable and are lost progressively throughout pregnancy. So this is the real challenge beyond 35. So preimplantation genetic testing for aneuploidy. It used to be referred to as PGS, or preimplantation genetic screening, but in 2017, the World Health Organization got together and harmonized terminology across obstetrics and gynecology, and so we all now refer to it as PGT-A. So over the last 20 years, there's been an intense amount of research, and it is confirmed that chromosomal aneuploidy is a major cause of IVF failure, as it is actually after natural conception, leading to pregnancy loss, as we've just seen, and in rare cases, abnormal pregnancies and affected live births. And the incidence of this in human eggs and embryos increases exponentially in women above the age of 35. And the progressive loss of these pregnancies with aneuploid embryos means that the incidence of recognized aneuploidies at birth is only between 3 and 5 of 1,000 births. So most of these embryos are not viable at all, and only a very small number reach full term. PGT-A, by next generation sequencing, really has given us a tool that allows accurate copy number analysis for all 24 chromosomes and at high resolution. And for that reason, PGT-A is now widely used as a highly effective additional tool for embryo selection. But this is almost always in combination with blastocyst culture, time-lapse or morphology scoring, biopsy at the blastocyst stage so that you can recover multiple cells, and then vitrification. which revolutionized the area because you can vitrify biopsied embryos, and there's still a very good viability. You don't affect their viability after thaw. And this is being done particularly for, in programs that want to focus on single blastocyst transfers. So what is the evidence for the efficacy of PGT-A? Well, when we're focusing on embryo selection, actually it is implantation rate per embryo transferred that we're interested in. And there have been several in the last 5, 6, 7, 8 years, mainly single center randomized controlled trials that have all shown improved implantation rates if euploid blastocysts are selected for transfer. Recently, a large multi-center randomized controlled trial, the so-called STAR trial, showed no overall difference with PGT-A in women aged between 25 and 40. So that's the young, good prognosis patients, as well as those in which the level of aneuploidy is much greater. But when the data was broken down, it did actually show a significant improvement in implantation rate per transfer in women between 35 and 40. And that's what's shown in this histogram on the right here, in which you can see that the addition of PGT-A has allowed the implantation, has restored, if you like, in this case, clinical pregnancy rates to the same level as women under 34. And this is actually significant and is quite a jump. So what does this look like? What does this testing look like? Well, first of all, I should explain these plots. And so the stripes here represent chromosomes 1 through to 22. So you might be interested to know that in the UK, because the HFEA does not allow us to reveal the sex of the embryo, the testing lab actually doesn't allow us to see the result for the X and Y chromosome, which would normally be here at the end of the plot. So what you can see here are 3 examples from one of our patients, actually, a 43-year-old patient, and on the left, chromosome 1 all the way through to 22, but not the X and the Y. The great thing about next generation sequencing is that you get a very good displacement. If there's an abnormal number of chromosomes, this is picked up because you find many more fragments that are sequenced. So there's random sequencing of hundreds of thousands of fragments from across the genome. And these are mapped back to their location on individual chromosomes and binned, so that these dots that are mainly running along the middle here actually refer to these bins. These are little segments of the chromosomes, and that is the normalized level of counts of fragments that have mapped back to those chromosomes. Now, if in the original biopsy and following the amplification, there was 3 or only 1 copy instead of the normal 2 copies, then you get many more fragments in that are being sequenced that map back to the affected chromosome if there's a trisomy, so you get this linear displacement. And similarly, if there's only 1 copy instead of 2, then you get this level here of only 1 copy. So there's a linear displacement, unlike the previous technology of array CGH. So you can see in all 3 of these embryos, unfortunately, there are full chromosome aneuploidies. Here a trisomy for chromosome 8, and here a monosomy for chromosome 21, and so on. Here a trisomy, here a monosomy, here a trisomy. But because we have this high resolution, we can also pick up if there are copy number changes for just part of the chromosomes. And so that's an example here of what we call a partial or a segmental aneuploidy. So this is quite powerful. And we now know that these can arise both during female meiosis, but also as the embryo is dividing. And so some of these are more serious than others. And as we'll see, the advice is if you're going to consider, in this case, as you can see, there are in fact whole chromosomal aneuploidies, so you wouldn't recommend this embryo for transfer. But if these weren't present, then you would have to, and you wanted to consider transferring this embryo, then we would recommend genetic counseling, because small changes in these chromosomes can actually lead to viable but affected pregnancies. And finally, we have the well-known changes that are intermediate between a full trisomy and a full monosomy, i.e. represented by these bars. And so you have these so-called mosaic changes, and these probably come about in most cases because one or more cells in that sample from the trophectoderm has actually got an aneuploidy, but you're mixing it with either normal cells or cells with different aneuploidies, and you can get some complex patterns. But again, bear in mind that in most cases, these are in the poorer prognosis patients towards 45 years of age, then you will normally find other aneuploidies. So, okay, high resolution NGS allows you to do whole chromosomal aneuploidy and partial or segmental aneuploidy, and you can pick up, because of the linear displacement, full or mosaic changes. So this has become hotly debated and has undermined the use of PGT-A. But recently, the International Society for PGD published guidelines that really can guide clinics what you should do in the case that you want to consider an embryo with just mosaic or segmental changes. So the first point I would make is that if within that cycle there's a euploid blastocyst, even if it appears to have a poorer quality, that should always be prioritized for transfer. And indeed, in our own experience at the Bridge Center, we have had pregnancies and live births from really quite poor quality blastocysts, which we've nevertheless biopsied. So you need a good biopsy practitioner, a good embryologist, who can do that without damaging the embryo. But we would always transfer that ahead of a good quality mosaic or segmental embryo. The second thing to bear in mind is that the incidence of these changes is actually not age related. So there's a level of mosaicism and segmental changes all the way through at all ages. And different data sets differ, but together they're about 10 to 20% per embryo. And this translates to those difficult cycles where you perhaps only have one of these to consider for transfer, might be 1 in 10 or 1 in 20. So it's not a huge problem, but obviously none of us want to discard an embryo that might be viable. And in those cases, the best practice is currently to offer the patient genetic counseling about the risks of those specific changes in those embryos, because we cannot at this time know whether or not that could in fact cause a serious abnormality. Now, clinical pregnancy rates following transfer of mosaic blastocysts is lower, and miscarriage rates are higher than for euploid blastocysts. And indeed, if you look at low levels of mosaicism and compare it with high levels or complex multiple mosaic changes, then the low level mosaicism are much more viable than the high level. And so I think it's very important to keep track of these and to offer genetic counseling if you can. Okay, so I'm now going to show you two sets of data. The first is from a clinic, actually several clinics in Southern California, led by Robert Anderson, who recently published their cumulative experience in a European Journal of Medical Genetics last year. So they've actually been using PGT-A for all patients for about 10 years and started with PGT-A, and they found it so beneficial that it changed because they had very good vitrification in place, it really revolutionized their ability to do single embryo transfers. And so they decided to change to PGT-A for all patients, irrespective of age or prognosis. And their experience with NGS-based testing over 6 years from 2012, actually some of this is OACGH, but it's mostly NGS, through to 2018 was published in this paper. I think so what I'm going to do with these results is actually to reprise the 2017 Japanese registry data for live births per ET. And remember, this is mostly, but not exclusively, single blastocyst transfers. And the same is true actually in Robert Anderson's clinic. They're mostly single blastocyst transfers, but not absolutely exclusively. So you can see here then the yellow bars are the live birth rates per ET in Japan. Now, when we consider the results from Bob Anderson's clinic, the implantation rates shown here in blue per ET are really incredibly high, and it was really their results that inspired us to develop our own single blastocyst transfer IVF program. So their average implantation rate per ET over a 6-year period with over 7,500 biopsied blastocysts was actually 75%, which I think you'll agree is really incredibly encouraging and very high. Furthermore, if they try to look at the live birth rate per cycle, so now taking into account all of those cycles that perhaps didn't get to egg collection, or if they did get to egg collection, they didn't get to the blastocyst stage, or if they got to the blastocyst stage, there were no euploids to consider for transfer, then of course the rate of live birth per cycle does come down, but remains incredibly high until about 40 years of age, and much higher than reported from Japan. As you get into women in their 40s, of course, this comes down, but it still compares very favorably with the rates in Japan, mostly without using PGT-A. So I now move to our own experience. So our focus was not in actually doing PGT-A for all patients, because the clinic that I worked in, the patients have to pay for their treatment. So we offered them the option of having PGT-A following genetic counseling. So all patients were counseled about their risk of having aneuploid embryos transferred, and we offered them optional PGT-A. But all patients of all ages, and they weren't a selected group, good and poor prognosis patients, were only offered single embryo transfer. And hence we called our program 1 by 1, I think for obvious reasons, to make it clear. So we tried to pull together all of the recent developments that we thought might help and might optimize and give patients the confidence to go ahead with single embryo transfer. So we performed blastocyst culture in a time-lapse incubator in low oxygen. We offered optional trophectoderm biopsy at that stage and NGS-based testing for PGT-A. And then all patients had freeze-all blastocysts by vitrification. And we were confident about going ahead with this program because already without PGT-A, our freezing program was giving us good rates of pregnancy. So then the patients had single vitrified warmed blastocyst transfer. We refer to this as VSET transfers in later managed cycles. And this was done for all patients. And importantly, within the package, again, encouraging them to go ahead with single embryo transfer and instead of, if you like, doing double embryo transfers, we included two frozen embryo transfers. And that's quite important because the clinical pregnancy rates I'll show you per ET, there are a few patients who have had 2 transfers and not just 1. So to recap then, in our program, embryo selection was by blastocyst culture. And of course, we know that by culturing to blastocyst, that improves pregnancy rates. For those patients that elected to have PGT-A, we selected euploid embryos that were clearly euploid. There were no mosaic and no segmental transfers in the data that I'm going to show you. And finally, we used manual time-lapse analysis of a cleavage pattern to select the best euploid embryo available for transfer. So the clinical work was published in 2019 in this paper here by Gorodecchia et al. And if you're interested in our approach to time-lapse, manual time-lapse analysis, then you can see the principles of that in this publication from McCollin et al. Okay, so these are our results. So let's go through this step by step. Again, I'm reprising the Japanese live birth rates per ET with the yellow bars here. And implantation rates now per VSET or single vitrified warmed blastocyst transfer are in the gray bars, and clinical pregnancy rates in the blue bars, and clinical rates per and delivery rates per cycle. And we kept, but this was a prospective cohort analysis, so we kept very careful records of all of the started cycles in all patients to get as close to what we believe would be our clinical pregnancy rate per cycle started in these patients. So as you can see, the first thing to point out is this first set of histogram bars. The only patients we found that actually elected not to have the PGT-A were patients under 35, presumably based on the fact that they have a lower chance of aneuploidy and a better chance of pregnancy overall. And so this data here are the results without, so these are single blastocyst transfers, and you can see that so we achieved very good implantation rates without PGT-A. And so this is based on, I think, perhaps the time lapse and the fact that we're doing the freeze-all strategy. In our hands, particularly in younger women, it's extremely beneficial and gives you very good pregnancy rates here. However, in those patients under 34 that decided to have PGT-A, then you can see that we had a staggeringly high implantation rate. So there's the number of patients. So I should have said that actually this is a very small data set, 106 transfers, of which and 200 patients, just over 200 patients, 263 cycles, and 15 second transfers. But in the patients under 34, there was approaching 30 patients in this group that had the testing. Almost all of them had implantation. And when you look at the clinical pregnancy delivery rate, similarly, it was over 90%. Now, of course, some of those cycles did fail for a variety of different reasons. And so when you look per cycle started, it does come down, but nevertheless is still well above 60%. Now, with increasing age, obviously, the rates do come down. And actually, there's a recent, and I can recommend, it's a paper by Scott's group that actually show that there are underlying problems in older women, even if you transfer euploid embryos. So our implantation rates came down, and of course, in parallel to that, the clinical pregnancy rates came down, but they were still extremely good. And our average implantation rate actually across the patients we looked at similar to Bob Anderson's group, were 76%. Now, of course, by the time you get into patients in their 40s, there are many cycles in which, for a variety of reasons, they may not have got to egg collection, as I said, and so on. And indeed, in some cycles, perhaps a third of cycles, there are no euploid embryos for transfer. So my advice with these patients is that it really is a question of finding that individual embryo that's euploid, because if you can find it, the pregnancy rates, even in those women, are really likely to be very good. And of course, the other side of the coin, and one of the major benefits of using PGT-A, is that the preclinical and post-clinical loss rates are low, in our case, 8 and 7% respectively. Okay, so then to summarize, what are the benefits of doing PGT-A? We all realize it has a cost associated with it. It does, it's labor intensive and so on at the moment, although there are exciting developments, as we'll see. But the first and most obvious one are the very high implantation and clinical pregnancy rates, combined with low miscarriage rates when you do this, even with single euploid vitrified warmed blastocyst transfers. And this has the effect of giving you a very sensitive way of deciding whether, in fact, an individual patient has underlying problems. For example, they may have a problem with an implantation window, and you then understand that if you do two sequential single blastocyst transfers without pregnancy in a younger woman, you know that there are other things that need to be investigated. So I would argue that it improves clinical management on the clinical side. And in terms of embryology standards, we've always looked to younger, good prognosis patients to benchmark our embryology programs. And of course, here it gives you a very good appreciation of the fact that everything has to be optimal, including, of course, embryo transfer. So you need to look at different clinicians and so on in order to get these very good results. Another benefit, particularly in younger women, is cryostorage of euploid blastocysts. So we have actually examples of patients. So this 3-year program has finished now. We're still following this cohort, and several women have come back having had a pregnancy and live birth for another transfer from that first cycle and have established a second pregnancy. And so this is very, very satisfying, not only for the patients, but also for the clinical team. And of course, for patients in their 40s that are facing miscarriage rates of perhaps 50 or 60%, then it really reduces that as a possibility. All right, so what would be my advice about incorporating PGT-A into clinical practice? Well, I think if your clinic already has experience not of PGT-A, but of PGT-M, which used to be called preimplantation genetic diagnosis, or is now called preimplantation genetic testing for monogenic disease, then all of this will be a lot easier. But my advice would certainly be, first of all, to take a good hard look at your pregnancy rates with single blastocyst transfers in a freeze-all setting. Do you have very good, robust vitrification? Because one of the key messages, I think, is that PGT-A will not compensate for poor clinical outcomes. So if you have rates that are currently low, then you really need to sort out your various procedures and protocols, both on the clinical side and the embryology side, before trying to bring in PGT-A. You certainly need experienced biopsy practitioners and embryologists to do the biopsy and vitrification. And so training is very important for that. And all of those needs to be optimal. And I would just remind you of comments I've already made that actually you need experienced embryologists, because any embryo that gets even to a poorer quality, obviously some blastocysts are barely cavitating and you can't biopsy them. But if biopsy is possible at all, you really need to do that in the older patients that are at very high risk of aneuploidy to find that one embryo. And if you can find that one embryo, it's become a mantra of mine in clinical practice, then we know that by transferring it, they have really quite a good chance of having a singleton pregnancy. You may need to consider if you need to set up a separate area and dedicated equipment in the embryology lab, and of course that takes time. But if you have a busy lab, a large lab, perhaps you have several manipulators already, and it may just be a question of dedicating particular equipment and so on, and sorting that out and working out how you're going to take on this extra work. And I would certainly recommend creating a core lead team with an experienced clinician, embryologist, and nurse. Nurses, I believe, are vital in this. They deal with the day-to-day communication with the patients. PGT-A is very complicated, and I think it's very important to do this. And I think the nurses enjoy this experience and enjoy being brought into the team. It's very important that they understand why we're trying to do this for these patients. And if you happen to have a molecular biologist or access to one, or a friendly lab director where you send your samples, that's extremely useful. And I would suggest having regular multidisciplinary review meetings and seeing where you are. Do we reach those benchmarks? And finally, I would say that genetic counseling is very important. This is not an add-on. In our program, we really believe in this. We were one of the first clinics to bring in a genetic counselor. Obviously, it's expensive to do that, but we were doing both PGT-M, single gene defects, as well as PGT-A. And I think the clinicians will very rapidly see the benefit of this because it's complicated, and patients need to be talked through the whole process and understand that at the end of the day, if they have this testing, they may not have an embryo for transfer, because otherwise the embryology team or the clinician will find themselves in a situation where the patients, feel that they haven't been adequately prepared for the whole process. So finally then, I thought I would end on an optimistic note in this difficult time. And I've picked up a number of future trends and developments and publications that I think are very exciting and mean that even today, with these kinds of results, I think there are still things we can do to improve our offering to patients of all ages, and both good and bad prognosis patients. The first of these, I think, is very interesting, and it's actually going back to using piezo-ICSI. And so a Japanese group actually looked at this, and they did a randomized trial in different women of different ages, and showed that in women over 35, they actually got improved fertilization rates and improved development through to blastocyst. And this may be correlated. We all know that in some women, the oolemma, the membrane around the egg, can be very fragile, and it can lead to, obviously, to when you do conventional ICSI, you can lyse the egg, whereas with piezo equipment, you can, it's so rapid, the forward movement is so rapid, and it's been used for years in animal species, particularly in the mouse, for example. The first ICSI all used piezo ICSI. And so this is very interesting, particularly in that group of women where we want to get just that one euploid embryo. So I think that's exciting. And furthermore, time-lapse incubators, yes, they are expensive and their capacity is improving all the time. And they have a particular benefit for programs doing embryo biopsy, because you can follow on a day-to-day basis exactly where the embryos are and pick a good time, because we know that obviously a cohort of blastocysts doesn't necessarily all get there on day 5. It could be to day 6, and even in some cases on day 7. So it's very useful not to have to open the incubator. And I'm just picked out one publication actually from the UK. It's a randomized trial that shows again that you can improve on the number of blastocysts you can get by culturing under these conditions. So combining these two approaches, I think will be wonderful if we can get more blastocysts. Then, of course, if you do have a time-lapse incubator, there are fantastic and very futuristic developments in artificial machine learning and artificial intelligence, and which can enable you to look at thousands of images. So you can imagine embryologists generally in the past have just looked at particular critical times and perhaps used a computer program to enable them to score embryos, but at critical times. Now with machine learning, you can look throughout the development of the embryo at thousands of images, and even we're beginning to look at different levels of focus, and which could never be done by eye. And so this is really going to fine-tune our ability to select the most viable embryos. And so if you combine that with finding euploid embryos, particularly in younger women, then I think that will be very exciting. And finally, of course, everybody is talking about the possibility of doing PGT-A non-invasively. And this is very exciting. Most people, although you can get, so about 10 years ago, it was discovered that you have some embryo-derived DNA in the culture medium. And this was quite a surprise, but perhaps it shouldn't have been, because we do know that all mammalian embryos undergo a certain amount of what is called apoptosis, or programmed cell death. And, you know, we weren't certain about where that DNA was going. And some of it appears to go into the medium. So this is very exciting. It also goes into the blastoceal cavity. So some people are proposing that, but I think most people are focusing, that's still invasive to some extent. So most, I think, people are looking at the possibility of using spent culture medium. And I just highlight two recent publications that actually show some very interesting data that demonstrate that the concordance can actually be remarkably good. The only caveat I would say, and much better than, I reviewed this in 2016 and have a publication of fertility and sterility if people want to look back. And I was a little bit, the jury was out, but I think we're moving towards it. But we will have to bear in mind that there are some cases in which there's no or very little DNA in the medium, and so we may still have to go back to do biopsies on some of the embryos. But let's see, I think it could be very exciting, and obviously could be one of the factors to improve the availability of this, which could be so useful to patients. So in my final slide, I'd just like to acknowledge the team that I worked with, Julia in particular, who led the clinical effort under Michael Summers's direction at the Bridge Center, Christian Ottolini, the senior embryologist, Jinjun Wang, who in fact was the head of the lab that we used, Sammy Newman, our genetic counselor, and Abia McCollin, and who was the data analyst, and Kamala Huja, who supported the work that we did. And here are all the references that I've referred to. So thank you very much. And I'll now answer one or two questions if I can.
Thank you so much for your presentation, Prof. Handyside. I think your insight into this technology is so valuable and especially interesting to hear from your own personal experience in clinical practice. We have a few questions if you are happy to answer them. So the first question is, based on the evidence that you presented today, would you recommend PGT-A to the age group under 34?
Well, certainly in our experience, we would recommend it. It's actually very good for younger patients. So not necessarily with a 35-year cutoff, but let's say 36, 37, 38, patients younger than that, it works extremely well and gives us exceptionally high rates. And you can't have experienced an IVF program that has those kinds of positive HCGs, and then you wait nervously to see if it's a viable pregnancy and so on, without feeling, that this is hugely beneficial. When you get into older patients, then of course the challenge is still very great, because sometimes you don't have those euploid embryos for transfer. So in the younger patients, what I would say is that some of those patients will have other underlying problems, and if you do PGT-A, then it's not aneuploidy that's causing the failures after transfer. And then you can focus more intensively on a whole variety of different clinical things that you could look at. And so for clinical management, I think it's actually still remains quite important. And the other wonderful thing, of course, is that these patients essentially, in many cases, even without overstimulating these younger patients, and of course by freeze-all, you remove the chance of ovarian hyperstimulation syndrome, they essentially have their family in the freezer. And it's so exciting for us to have second pregnancies from one cycle. I know it's, of course, some good clinics are doing it every day, but it still is very satisfying for both patients and the clinical team.
I can see how that would be very exciting for the clinical team. So the other question that we have is, how should mosaic embryos be used in a clinical setting?
Well, I think the truth is that we're still at very early stages. People are collecting data. So what clinics are doing is, generally speaking, if patients have a cycle of treatment and there's only a mosaic embryo or an embryo that has a segmental error, they're getting patient consent to transfer the embryo following the genetic counseling. So I think it's very important that you put counseling in place, because otherwise you risk an abnormal pregnancy, which wouldn't be good for anybody. Now, the data from the New England Journal paper by Greco et al. A few years ago did demonstrate that mosaic embryos can be viable. So they can and should be considered for transfer, but only if you don't have a euploid embryo. And then there seems to be an emerging consensus that low-level mosaics are probably just a low level of aneuploidy caused by abnormal segregation in the embryo. We know that there are many abnormal divisions that occur as the embryo develops over the first week, and that they might reduce the viability, but they're not clinically significant. And so it depends on the level of mosaicism, and I would warn against, transferring high-level mosaics, because those could have meiotic aneuploidies, which will affect the whole embryo and then result in a miscarriage.
Thank you. I think this is something that we all find really tricky, and so good to have your insight. The last question that we have is, have you seen evidence of culture conditions influencing the aneuploidy mosaic rates?
Well, as people will be aware, some people have suggested larger labs have looked at their associated clinics and asked them questions about what their culture media, and have shown some interesting differences. So there are apparent differences between clinics, so we can't rule this out. And I'm particularly interested still on a research basis. Embryos have a lot of abnormal divisions, and it's the abnormal divisions we were looking for to select embryos, so that if an embryo has an abnormal division early in cleavage, then it can scatter the chromosomes in the daughter cells, and which are completely not viable, and so on. So it's an interesting question, and I think is for future research. Can we, in different culture conditions, reduce the level of those abnormal divisions and so on? And to what extent are we causing some abnormalities in the embryo? And I think, again, there's still a lot to be done.
Thank you so much. Thank you for answering all our questions, Prof. Handyside. It doesn't look like there are any more that have come in. It really has been a privilege having you present to us today. Thank you to everybody for joining us, and please do keep safe. And I think we'll end the webinar there. Thank you so much.
Thank you.